forget your backpack.
Oh yeah.
Let's go! Jump in!
Vámonos!
You can lead the way! Hey!
Today we're going to teach you all about the sliding muscle filament theory. In Spanish, it's del mente de teoría del filamento muscular. Can you say that in Spanish, Brendan?
No, I can't.
Okay, well, I guess it's not really important.
Alright, let's talk about the adventure we're going to go on today. We're going to learn about the sliding muscle filament theory.
But first, we should probably cover the basics, right?
Yeah, alright. Let's
Alright.
show them. Can you find an example of a muscle that can contract and relax?
Backpack!
What was that?
Backpack! Have to click on backpack. Click backpack. Click backpack. Backpack. Yeah!
Check Dora's backpack to find the muscle that can contract and relax. That's right, the bicep brachii.
Skeletal muscles like the bicep brachii are attached to bone via connective tissue called tendons. Muscles are composed of bundles of muscle fibers. Each bundle is separated by connective tissue known as perimysium. Each smaller strand is made of muscle fibers, which are separated by connective tissue called endomysium.
Skeletal muscle fibers are cells multinucleated and striated in appearance. Muscle cells are composed of subunits called myofibrils. Each myofibril is made up of several myofilaments. The two types of myofilaments shown in red are composed primarily of the protein myosin, and a thin myofilament shown in blue composed mainly of the protein actin. The repeating arrangement of the thick and thin filaments serves as a fundamental subunit of striated muscle contraction. These subunits are called sarcomeres. Sarcomere contraction is represented by the shortening of the distance between the Z lines, which is 2/3. The sarcomere shortens because the thin filaments slide past the thick filaments. In 3D, each thick myofilament is surrounded by 6 actin filaments.
All these microscopic steps contribute to the sliding muscle filament theory.
Muscle contraction occurs when a wave of positive energy innervates the motor unit, also known as the neuron cell body. Let's pretend that we're the action potential. First, we would start in the motor unit, then we would travel down the axon. When we get closer to the muscle fiber, we would split into terminal branches. When the terminal branch attaches to the muscle fiber, this is known as the neuromuscular junction. Let's take a closer look.
This is a close-up of the neuromuscular junction. The action potential travels down the axon into the axon terminal. The action potential releases acetylcholine, which is a neurotransmitter.
Acetylcholine travels into the motor end plate and is accepted by receptors. The receptors accept the acetylcholine and send an action potential back out. Since we're the action potential, where would we go next?
Who can we ask for help when we don't know which way to go?
If there's a place you want to go, I'm the one you need to know. I'm the map, I'm the map, I'm the map!
A map!
You tell Dora she needs to travel across the cell membrane and then down the T-tubule. Across the cell membrane, now down the T-tubule. Next, the action potential will stimulate IP3, which will allow for the release of calcium out of the sarcoplasmic reticulum. When calcium is released, it will next enter the sarcoplasm.
The formation of a cross bridge is initiated when calcium ions are released from the sarcoplasmic reticulum. Calcium binds to troponin. Troponin then changes shape. Troponin ensures that tropomyosin moves off the actin binding site.
This allows the globular head to attach and form a cross bridge.
Sorry, coming through.
Oh, no. Nothing gets past my body.
Ugh. Hey! Cash money.
Okay.
Sir?
Now you can go in.
Also note that the globular head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the globular head. ATP is then hydrolyzed into ADP and inorganic phosphate.
The energy created from the hydrolysis of ATP then activates the globular head, forcing it into the cock position. Cross-bridge cycle may be divided into 5 steps. Step 1. Relax muscle. The cross-bridge is detached. However, it is in a high energy due to the previous hydrolysis of ATP. It is now ready for action.
Step 2. Cross-bridge formation or attachment. The activated globular head binds to actin, forming a cross-bridge. The inorganic phosphate is released, and the bond between myosin and actin becomes stronger.
Step 3. The power stroke. ADP is released, and the activated globular head pivots. This then propels the actin filament. Step 4. The shortest step, Ricker complex. The cross bridge is temporarily stuck to the actin.
Step 5. Cross-bridge detachment, or release. ATP binds to the globular head. The link between them weakens the globular head and allows the globular head to detach from the actin. ATP is then hydrolyzed into ADP and inorganic phosphate. The energy release during the hydrolysis reaction reactivates the globular head, returning it to the cock position, ready to repeat its cycle. Finally, an ATP-driven calcium pump is responsible for pumping the calcium out of the sarcoplasm back into the sarcoplasmic reticulum.
We did it!